Identifying the specific sugars hidden within a sample, whether it's glucose, fructose, or starch, requires monitoring how individual molecules respond to chemical reagents through distinct color changes. The first step in this process is Molisch's test. This serves as a universal baseline, confirming the presence of any carbohydrate in the sample before we move to more specific identification. Precision in physical technique is required here. Concentrated sulfuric acid must be poured slowly along the inner wall of an inclined test tube, ensuring the layers do not mix. As this chemical diagram illustrates, D-glucose reacts with the acid and alpha-naphthol to produce a condensation product. When performed correctly, a violet or purple ring will form precisely at the interface of the two liquids. While this purple ring proves a carbohydrate is present, it does not distinguish between types. To isolate the exact molecule, we must apply more specialized tests. With the presence of a carbohydrate confirmed, we now need to determine its structure, specifically whether it is a simple monosaccharide or a disaccharide. For this classification, we use Benedict's and Fehling's tests. These are designed to identify reducing sugars, which are carbohydrates that possess a free aldehyde or ketone group. This equation shows the Benedict's test mechanism. In an alkaline medium, the reducing sugar converts copper-2 ions into copper oxide. The resulting chemical reduction is visible as a solid brick red precipitate that drops out of the solution. If you observe this red precipitate in an alkaline environment, you have definitively isolated the reducing sugars from the rest of the sample. Tollin's test provides a secondary method for detecting these reducing sugars. Instead of copper, this reaction involves the reduction of silver ions into metallic silver. Here, silver oxide reacts with glucose to yield gluconic acid and pure silver. This metallic silver deposits onto the inner walls of the tube, creating a visible silver mirror. Since these tests rely on the presence of a free aldehyde or ketone group, consider the behavior of sucrose or starch. Based on that structural requirement, would you expect these samples to produce a red precipitate in a Benedict's test? They will not. Both will yield a negative result because their chemical bonds lock the functional groups required for reduction. Sucrose is a non-reducing sugar, and starch is a complex polysaccharide. Because they lack those free groups, they consistently remain unreactive in these specific alkaline tests. To identify the complex polysaccharides that did not respond to the reducing sugar tests, we use the iodine test. According to these notes, starch reacts with iodine to form a deep blue-colored solution, which disappears when heated and reappears upon cooling. The test also differentiates glycogen, producing a reddish-brown color, and dextrin, yielding a brown solution. These specific color variations allow for the confirmation of complex structural carbohydrates that otherwise appear unreactive. To manage the findings from all 5 biochemical tests, we can organize them into a single observation table. The Molisch row demonstrates that every carbohydrate in our set, from simple glucose to complex starch, yields a positive purple ring. In contrast, the Fehling, Benedict, and Tollin rows show positive results only for the reducing sugars. Sucrose and starch are marked negative here. Finally, the iodine row isolates starch with a blue-black result, while all other columns remain negative. You can use this completed matrix as a reference for your practical work. Understanding how these results overlap provides a systematic way to identify unknown carbohydrates in the laboratory.